Hi, my name is Sergiana, and I'm here to present you beta thalassemia. So I'm here to talk about this kind of disorder. At least you have a knowledge of what it is about. Okay, so for the next page, what is beta thalassemia? Beta thalassemia is a type of blood disorder that reduces the production of hemoglobin. Hemoglobin is the most important protein in our body that is in the red blood cells that carries the oxygen from the lungs to the body's tissues and returns carbon dioxide from the tissues back to the lungs. So when there are low levels of the hemoglobin, it can lead to a lack of oxygen and in many parts of the body. So the thing with this kind of disorder, it causes anemia, and it makes the person more fatigued. So with the beta thalassemia is involved with the beta globin gene that reduces the production of hemoglobin. So there are three types of the beta thalassemia. We have the beta thalassemia major, intermedia, and the dominant beta thalassemia. So the mutations in the HBB gene is a main cause of this type of disorder. There are two subunits in the beta globin. So we have a beta 0 and a beta plus. So the mutations that prevent cells from producing any beta globin results in a beta 0. The HBB gene mutations that decrease the beta globin production occurs in the beta plus. So there are several mutations that insert or delete a small number of nucleotides in the HBB gene. So what are the common signs and symptoms of this disorder? We have fatigue, jaundice, weakness, slow heart rate, facial bone deformities, slow growth, abdominal swelling, dark urine, splenomegaly, and weakness and fatigue. So here we see a picture of what a normal red blood cell will look like, and then a malformed red blood cell from thalassemia. So when we see here of a normal red blood cell, it's like a biconcave type of shape, like a circular shape, that transports the oxygen to all parts of the body's tissues. But with thalassemia, as you see here, it looks dysfunctional, it's malformed, misshaped, and it actually makes the spleen work harder just to recycle those kind of blood cells. Prevalence and responsible gene and the chromosome location. So beta thalassemia is not very common. It's kind of rare in the United States. There's like an estimate of approximately 1 out of 100,000 individuals in the area that have the symptomatic cases. Normally with this kind of disorder is passed from parents to children and is common in Mediterranean countries like in Greece, Southeastern Asia, Middle East, and then some African countries. So I know I mentioned earlier in the other slides that this kind of disorder is caused by mutations in the DNA of the genes that make the hemoglobin. The mutations associated with thalassemia are usually passed from parents to children. So most beta thalassemia cases are caused by mutation in the HBB gene, and this type of gene creates the beta globin protein chains. So the mutation in two HBB genes can reduce the levels of beta chains. So with two mutated genes, the thalassemia signs and symptoms will be mild. So this is referring to like an alpha thalassemia trait. And when you have one mutated gene, usually there's no signs or symptoms of thalassemia. And then with the three mutated genes, there are a lot of signs like moderate to severe. So what is the mode of inheritance for this type of disorder? So it's inherited in an autosomal recessive pattern. And usually with this kind of recessive disorder, an individual will inherit an abnormal gene from each parent, unfortunately. So the parents of the individual with this autosomal condition at least each carry one copy of this mutated gene, and they typically do not show the signs and symptoms of this condition. So individuals with only one HBB gene mutation in each cell develop a mild anemia. So these mildly affected people are said to have the thalassemia minor. Here I put like a Punnett square example of a couple who are afflicted with the beta thalassemia. So here we see the keywords, the genotypes with the letters, and the phenotypes of the characteristics. So the double B, capitalized B, stands for normal. Capitalized B and lowercase b is the beta thalassemia carrier, and the lowercase double b stands for the disorder. So say that you have both parents who are afflicted with the disorder. So what is the chance that the offspring will have this kind of disorder? So there might be like a 25% chance or 1/4 probability that the offspring will have the disorder, or maybe a 25% chance that the child will be normal, or a 25% chance that the offspring will be a carrier. So here is a pedigree diagram example with 4 generations. So I'm just going to focus on the 4th generation, the children here as you see. So for instance, you see that FT and FI are the offsprings of FC and FN. So FC, who's the mother, does not have the disorder, so she's normal, and FN has a disorder. So as you see here, the daughter Fi is normal and FT is a carrier. But FT does have other cousins who are afflicted with this disorder, like SS, DI, and SA. So here's just a little brief description of, this is like a generation of a Romanian family. So it's common for them to have this kind of disorder in the family. So there are available genetic testing options for this kind of disorder. We have beta globin, HBB sequencing, is a procedure that reads instructions of the DNA that make up the HBB gene. And this kind of test identifies the presence of like a malformed, altered HBB gene in the person's cells, which may help a doctor to confirm the diagnosis of this beta pathology, hemoglobin pathology. This kind of test deals with the sequence analysis of the entire coding region. Then we have the deletion duplication analysis, as you see here. This kind of test finds out whether the HBB gene contains mutations resulting from the presence of duplicated or deleted parts of the DNA code. And now we have the prenatal and pre-implantation genetic testing. So this kind of testing tests for both high-risk pregnancies and intermediate risk pregnancies, and is recommended for single gene testing. But what is the typical cost for genetic testing? Normally it runs around $640, which is a pretty high number. There are a few companies that offer this kind of testing. You have the ARUP laboratory, good for sequencing tests, and then you have the LabCorp and then Prevention Genetics. So I put here about this information of technology, like several like current and then future therapies to treat the disorder. So like for instance, like for few patients, current therapies include a lifelong monthly supportive red blood transfusion of the iron chelation, and with some drugs such as the deforaxmin, deferprone, and then the defersorox. And then you see there's gene and cell therapy, and there's the allogenic HSC transplantation, and we have the pharmaceutical induction of the gamma globin. So the pharmacological induction of the fetal hemoglobin and gene therapy are currently at the experimental stage. And then that's it. This is all the information you need to know. So here are just some references if you wanted to learn more about this disorder. Thanks for watching.